Hello, my name's Cameron Burke, and welcome to this Kaplan MCAT Science Review for Biosignaling. This video covers the different ways biosignaling goes on in your body, including the role ion channels play, as well as their slightly more complex biosignaling counterparts, the enzyme-linked receptors and G-protein-coupled receptors. The goal to shoot for by the end of this video is to be able to identify what the major cellular signaling processes are and what proteins play an important role in these processes. At the end, you'll be able to test your progress on questions like this one about protein regulation of ion concentrations. Ion channels are vital for biosignaling because they allow for the movement of molecules that otherwise couldn't deliver messages due to charge. On the MCAT, there are three broad categories of ion channels you should know about: ungated ion channels, voltage-gated ion channels, and ligand-gated ion channels. The most basic type of ion channel is the ungated ion channel. These guys are always open, and their specific ion is allowed to diffuse across the membrane whenever it wants, or whenever the ion gradient demands it. A classic example you could be asked about on test day is the potassium channel, which is in every cell and allows potassium to flow in and out of the cells as needed. Unlike ungated channels, voltage-gated channels are slightly more particular in that they only open at a specific cell potential. A charge differential in the membrane potential opens the channel, then ions redistribute along their gradient, and once the differential is not the exact differential needed to trigger the opening, the channel closes again. One example of this phenomenon is the repolarization of neurons after an action potential. Potassium molecules are stabilized in the channel by water molecules, and are only allowed through when the membrane potential causes the channel to open. Our last type of channel is the ligand-gated channel. Of the three types of ion channels, ligand-gated channels are the most specific about the circumstances under which they'll open. Before any ions can pass through, a specific ligand molecule, like a hormone, binds to a receptor site and allows the gate to open. Once the ligand binds and the gate opens, only then can ions pass through the channel. One example you might see on the MCAT is the GABA receptor channel, wherein the binding of GABA to its channel allows for the flow of chloride ions to hyperpolarized neurons, acting as an inhibitory neurotransmitter. In general, ligand-gated channels play many roles in the nervous system. Ion channels play an important role in biosignaling by allowing ions to travel through them to deliver messages, but many times biosignals can be sent through surface receptors, like an enzyme-linked transmembrane receptor, where the receptor sends the message following surface binding, rather than a molecule traveling through the cell itself. As just a general structural outline, enzyme-linked receptors are two separate enzymes that each have three sections, including the ligand-binding domain, the transmembrane domain, and finally the catalytic domain, where the actual reaction is catalyzed. A ligand comes in to bind, and the two ligand binding domains close around it, causing a conformational change in both transmembrane domains that triggers the catalytic response necessary to deliver a signal. G-protein coupled receptors are much like enzyme-linked receptors in that they need a ligand to bind outside the cell, but where the message goes from there is slightly more complicated than a simple transmembrane protein. This is one example of a generic GS protein. The ligand binding site itself is attached to 7 transmembrane alpha helices. The receptor subunit is bound to an alpha subunit of a G protein on the inside of the cell, which usually binds GDP. When the ligand comes to bind, a conformational change occurs which causes the subunit to release GDP and bind GTP. G-protein S is responsible for increasing cyclic AMP, initiating signal cascades. When a ligand causes GTP to bind instead of GDP, the alpha subunit brings GTP to adenylate cyclase. GTP releases a pyrophosphate and becomes GDP, activating adenylate cyclase. A molecule of ATP is converted to cyclic AMP and a diphosphate, initiating the signal cascade, and the alpha subunit brings a GDP back to the G protein. G protein S is the most relevant for increasing biosignaling, but there are actually 3 G proteins you could potentially be asked about for the MCAT. GS protein is the first one. It stimulates adenylate cyclase and increases cyclic AMP, turning on signal pathways. The second is GI protein, which inhibits adenylate cyclase, decreasing cyclic AMP and turning off signaling pathways. And the final type of G protein that should be your main concern is GQ protein, which doesn't deal with adenylate cyclase at all, but rather works on phospholipase C to increase signaling molecules. These are really 3 examples of G proteins, but there are many more that operate in eukaryotes. Regardless of which G protein you're specifically thinking of, they all play important roles in influencing biosignaling pathways by increasing and decreasing the amounts of chemical messages that are sent around the body. Now you have a chance to practice and assess your progress with a learning goal. The first practice question refers to action potentials in signaling. The correct answer is C. Elimination is the best way to get through this question. You can get rid of anything that is going to work for the nervous system. In general, ions play a super important role in the nervous system because they help make a charge gradient, so you can cross off A, B, and D. G protein coupled receptors are the only possible choice that must rely on a ligand and not on a difference in potential to be activated. The second practice question deals with cyclic AMP activation. D is the right answer. G has proteins responsible for stimulating production of cyclic AMP. To recap this review, there are three types of ion channels that play a role in signaling: the ungated channel, which is always open, the voltage-gated channel, and the ligand-gated channel. Receptors also play important roles in biosignaling. The first type of receptor is the enzyme-linked receptor, and the second type is the suite of G protein-coupled receptors, including the GS protein. To turn on signaling, G protein-coupled receptors send GTP to adenylate cyclase to increase cyclic AMP. But there are many other types of G proteins with different jobs you might need to know about on the MCAT. You made it! Thanks so much for watching this Kaplan MCAT science review about biosignaling, and good luck on this topic on test day.